You know, usually when we talk about our DNA, we talk about it like it's a software program. Like we picture this sequence of letters, you know, A, C, T, G, just sitting there on a hard drive waiting to be read.
Right, like static code.
Exactly. But today, for this deep dive, we're going to completely flip that script. I want you, the listener, to imagine your DNA not just as this static genetic code, but as a physical, three-dimensional architectural structure.
Yeah, that 3D spatial element is huge.
It really is. Imagine it has scaffolding, anchor points, and like load-bearing walls. Because the mission for our deep dive today is to explore this microscopic architecture.
And it's a phenomenal structural problem when you really think about it.
Oh, totally. We're going to figure out how a cell manages to pack 2 whole meters of DNA into a microscopic nucleus without it just turning into a chaotic, tangled mess. And maybe more importantly, we're going to see what happens when you take a sledgehammer to those load-bearing walls.
Yeah, and to understand how the cell actually solves this packaging problem, we are going to be relying on a truly groundbreaking piece of source material today.
Let's hear it.
It's an article from Nature Cell Biology, published in February 2026, and it was led by a team of researchers at ETH Zurich.
Okay.
The paper is titled LBR and LAP2, Mediate Heterochromatin Tethering to the Nuclear Periphery to Preserve Genome Homeostasis.
That is a mouthful.
It is a bit dense, yeah. But the implications of this research are just profound because, as these researchers discovered, the physical geographical location of your DNA inside the cell is quite literally a matter of life and death.
Okay, let's unpack this because before we can start swinging sledgehammers and, breaking the cell's architecture, we need to understand how it's built in the first place.
Exactly.
You've got this tiny nucleus, and the conventional layout of a cell's nucleus across almost all eukaryotes is incredibly organized, right?
Very much so.
It's not just a bowl of spaghetti. There's a very specific geography to it.
There is. And if you were to peer inside a healthy nucleus, you would see this very distinct spatial segregation.
Okay, paint a picture for us.
So the active DNA, the genes that the cell is actively reading and using to build proteins right this second, that sits right in the middle of the nucleus.
Okay.
We call this active open DNA euchromatin.
Euchromatin, got it.
Right. But then you have the inactive silent DNA. These are the genes the cell doesn't need, or maybe the ancient, potentially dangerous sequences it wants to keep permanently shut down.
The stuff it wants to hide.
Basically, yeah. This silent DNA is called heterochromatin, and it is physically pinned to the outer edges of the nucleus, right up against the nuclear envelope.
I love visualizing things. So let's use a mental image to really lock this in for you listening. Think of the nucleus of your cell like a massive ancient library.
I like that analogy.
Right. So the frequently read, highly useful books, the euchromatin, those are left wide open on tables right in the center of the reading room. Anyone can walk up, browse them, and use them to get work done. But the dangerous, obsolete, or, restricted books, the heterochromatin, those are slammed shut, padlocked, and shoved into bookshelves that are bolted tightly against the outer walls of the library.
That library analogy works beautifully. But to make it even more accurate to the actual biology, imagine that the library shelves themselves are almost like magnetically binding those restricted books to the wall.
Oh, wow. Okay.
Yeah. Those bookshelves at the walls have a scientific name. They're called lamina-associated domains, or LADs.
LAs, LADs.
Correct. And these LADs are massive structural features. We are talking about regions of DNA up to 10 megabases in size.
Wait, 10 megabases? So that's 10 million DNA letters just locked up in a single stretch.
10 million. They are entirely gene poor, meaning there really aren't many active instructions there anyway.
Okay.
But to make absolutely sure these restricted books aren't accidentally opened and read, the cell covers them in specific chemical do not read signs.
Like actual chemical tags.
Exactly. And the most prominent of these chemical signs is an epigenetic mark called H3K9A3.
H3K9A3. That sounds like a Wi-Fi password.
It does, yeah.
Let's break down what that actually is and how it physically works in the cell, because we want to explain every detail here.
Okay, so it sounds like a password, but it's actually a physical modification. Inside the cell, your DNA isn't just a loose thread.
Right.
It is wrapped tightly around these tiny protein spools called histones. This is what keeps it organized.
Like thread on a spool.
Exactly. And when the cell wants to lock a section of DNA down, it adds 3 specific chemical tags, methyl groups, to a very specific spot.
Okay.
Specifically to lysine 9 on histone number 3. hence H3K9Me3.
Oh, I see. H3 is the histone, K9 is the spot, and Me3 is the 3 methyl groups.
You got it. And when those methyl groups are attached, it causes the DNA to spool up incredibly tightly. It compacts it so densely that the cellular machinery physically cannot get in to read the genes.
Like pulling a zip tie closed.
Yes, exactly like a zip tie. And what's staggering is that up to 40% of your entire genome is locked up this way and pushed to the periphery in these lamina-associated domains.
Wait, 40%.
Yeah.
Almost half of all the DNA we possess is just bolted to the walls of the nucleus and completely shut down.
It is a massive architectural feature. I mean, it takes a tremendous amount of cellular energy to maintain this structure.
Okay, so before we move on, let's just do a quick recap for you listening to make sure we've got the foundation locked in.
Good idea.
Number 1, the active DNA, the euchromatin, sits in the middle of the nucleus like open books. Number 2, the silent DNA, the heterochromatin, is shoved to the edges like locked bookshelves.
Mm-hmm.
And number 3, this whole organization is maintained by physical tethers and chemical zip ties like H3K9E3.
Perfect summary.
So here is where my mind immediately goes. If up to 40% of our genome is physically bolted to the walls, what are the actual screws holding it there?
Right, what's actually attaching it?
Exactly. How is it physically attached? And because scientists love to break things to see how they work, what happens if we unscrew them?
Well, that is exactly the driving curiosity behind the ETH Zurich researchers project. They wanted to find the exact structural screws holding the silent DNA to the wall.
Okay.
Now, previous studies had already given the scientific community some clues. Biologists knew about a structural meshwork inside the nuclear envelope called the nuclear lamina.
The nuclear lamina?
Yeah, which is basically made of proteins called lamin A and lamin C. They also knew about a protein embedded in the membrane called the Lamin B receptor, or LBR.
Okay, so you have the lamin AC mesh and the lamin B receptor. So the obvious next step for an experiment is to just remove those and watch the whole thing come crashing down, right?
You would think so.
Oh.
But here was the really frustrating part for early researchers.
Oh.
When they went into mammalian cells and genetically deleted lamin AC and that lamin B receptor, the library shelves didn't collapse.
Wait, really?
Yeah, the silent DNA mostly stayed stubbornly attached to the walls.
That is wild. You take out the suspected load-bearing pillars and the roof just stays up. How is that even possible?
It points to a massive biological redundancy.
Ah, backups.
Exactly. The cell has evolved backup system upon backup system to keep that DNA at the periphery. The cell kind of knows that letting this DNA loose is catastrophic, so it doesn't rely on just 1 or 2 tethers.
That makes sense.
So the ETH researchers realized they couldn't just knock out 1 or 2 proteins. They had to launch a massive coordinated strike against the entire internuclear membrane to overwhelm all those backup systems.
A coordinated strike. I love that.
Yeah, and they used a technique called reverse genetics.
Reverse genetics. Okay, let me make sure I'm following this. Normally, genetics works by finding a sick cell or, a mutated organism and then working backward to find the broken gene that caused the problem.
Right, that's forward genetics.
So reverse genetics is the opposite. You deliberately break specific, known genes and then sit back and watch what happens to the cell to figure out what those genes were actually doing.
You have the concept down perfectly. So to execute this, the researchers selected 12 different highly abundant proteins that live in the internuclear membrane.
12 of them.
Yeah, they suspected the master screws were somewhere in that lineup. But to knock all 12 out simultaneously, because remember, they have to overwhelm the backups they had, to use a highly specialized tool called CO pools.
Okay, CO pools, SI pools. Explain the mechanism here. How do you simultaneously turn off 12 different proteins in a living cell without just killing the cell instantly?
It's actually really clever. It relies on intercepting the cell's internal mail system.
Okay.
So think about the central dogma of biology. DNA makes RNA, and RNA makes protein.
Right.
The DNA is the master blueprint locked in the vault. The RNA is the photocopied instruction that gets sent out to the cellular factory to actually build the protein.
Okay, tracking.
Well, SIPOLs are essentially pools of small interfering RNAs. They are custom-designed molecules injected into the cell that act like mail interceptors.
Oh.
Yeah, they hunt down the specific RNA photocopies for those 12 membrane proteins and destroy them before they ever reach the factory.
Oh, that's brilliant. So the cell's DNA is still trying to send the instructions to build the walls, but the instructions are being shredded in transit.
Exactly.
So the factory stops building the 12 proteins, and eventually the existing proteins just naturally degrade and disappear.
Yes. And they tested this on human colon cancer cells, known as HCT116, and also human retinal cells, called RPE1.
Okay, so a couple different cell types, to be sure.
Right. They hit them with disciples, intercepting the blueprints for all 12 proteins at the exact same time, and total structural collapse.
Total collapse. I actually looked at the microscopy images from the paper, and the visual is striking.
Isn't it?
It really is. For the listener, they used a green fluorescent dye to tag that H3K9A3 password, the tightly spooled silent DNA we talked about.
Right.
Normally, it looks like a neatly drawn, bright green circle outlining the very edge of the nucleus. But after the psi-pools hit, that ring completely vanishes. Instead, all that green restricted heterochromatin detaches from the walls and clumps together into large, floating, glowing blobs right in the middle of the nucleus.
Yeah, the imagery is wild.
It goes from looking like a perfect circle to looking like a lava lamp. The library shelves had finally fallen over.
The visual evidence was undeniable. But of course, this creates a completely new problem for the researchers.
Right, because they knocked out 12 proteins to get this collapse. 12 missing screws is a lot.
Exactly.
If you take 12 random bolts out of your car and the engine falls out, you still don't know which specific bolt was snapping the engine block up and which ones were just, holding on a plastic cover.
That's a great way to put it.
So how did they narrow down the suspect list?
Well, they had to play this really painstaking game of biological elimination. They call it deconvolution.
Deconvolution.
Right. They started with cells that were already missing the foundational lamin AC proteins. Then, instead of knocking everything out, they systematically added the other suspects back into the cell, group by group, combination by combination.
Oh, man.
Yeah, they were looking for the absolute minimum requirement, the smallest combination of missing proteins that would still cause the heterochromatin to detach.
So by putting the parts back one by one, they could see exactly which pieces actually restored the structural integrity of the wall. That must have taken forever.
Oh, it is incredibly labor-intensive. But through this process, they arrived at a massive breakthrough.
What did they find?
They discovered that to force a total detachment of the heterochromatin, you only really needed to remove 3 specific things.
Okay.
Lamin AC, the Lamin B receptor we mentioned earlier, and a protein called LAP2, and specifically a version of it called the LAP2 beta isoform.
LAP2. Okay, that's a new character in our story. Where does it come from?
LAP2 is part of a family of proteins that interact with the internuclear membrane. It's encoded by a gene called TMPO.
TMPO, got it.
So when the researchers knocked out LBR and the TMPO gene in cells that already lacked Lamin AC, the heterochromatin collapsed entirely. Out of the massive lineup of suspects, these 3 proteins are the master keys. They are the primary load-bearing structural screws holding the library shelves to the walls.
Okay, let's do another quick recap for the listener to track the demolition phase here.
Let's do it.
The cell uses a highly redundant backup system to keep its silent DNA pinned to the walls. By using small interfering RNAs, the psi-pools, to shred the blueprints for 12 proteins at once, researchers bypassed those backups and caused a total collapse.
Right.
And then through a process of elimination, they proved that LBR, LAP2, and Lamin AC are the critical tethers.
Perfect.
But, this brings up a huge question for me. If the cell spends this much energy and builds this many redundant backup systems just to keep this DNA bolted to the wall, what exactly is it so afraid of?
There's a big question.
Like, what actually happens when those restricted books finally spill out onto the center tables?
Well, this is where the consequences of the structural collapse become truly dramatic.
Yeah.
Because physical detachment isn't just a matter of the DNA changing its ZIP code inside the nucleus.
Right.
The physical location dictates the chemical reality.
So moving away from the wall actually changes the DNA itself.
It changes the packaging. When the heterochromatin falls away from the nuclear periphery and flows into the active center, it starts losing its chemical locks.
The zip ties.
Exactly.
Yeah.
Remember the H3K9V3 password? The researchers found a significant reduction in that mark, as well as another repressive epigenetic mark called H3K27E3.
Oh, wow.
Yeah. Without the physical tether to the nuclear envelope, the chemical silencers just start to naturally degrade and fall off.
Okay, and here's where the paper takes a wild turn. Because when the DNA detaches from the wall and loses those chemical locks, the cell suddenly hits the panic button.
It really does.
It starts acting like it has been infected by a virus. It triggers this massive antiviral innate immunity response.
Yeah, so when they ran the RNA sequencing to see what the cell was doing, the data showed this huge spike in genes related to the interferon response.
Wait, interferon, that's a viral alarm, right?
Exactly. Interferons are essentially the cell's frontline defense, or alarm bells, against viral infections. They basically tell the surrounding tissue, we are under attack, shut everything down.
But wait, I need to pause here because this makes no sense at first glance. We didn't infect the cell with anything?
Nope.
The researchers didn't introduce a virus in a petri dish. They literally just broke a few protein screws and moved the cell's own DNA from the edge of the nucleus to the center. Why on earth does the cell's immune system think an external virus has invaded?
To understand that, you have to look very closely at what is actually written inside those restricted library books.
The so-called junk DNA.
Exactly. We often hear the term junk DNA, but a huge portion of that junk isn't just random empty filler. It consists of ancient viral remnants and parasitic jumping genes known as transposable elements, or transposons.
Jumping genes. That sounds vaguely ominous.
It is a little ominous. Think of elements like lines, long interspersed nuclear elements. Over millions of years of mammalian evolution, ancient retroviruses infected our ancestors, and they survived by reverse transcribing their own viral RNA into our human DNA.
So they literally copied and pasted their genetic code into our genome.
Yes. Over generations, many of these got trapped and mutated, but they still retain the basic machinery to copy themselves and jump to new locations in the DNA.
So we are essentially walking graveyards for ancient viruses. Our DNA is just littered with the corpses of infections from millions of years ago.
That's a very accurate way to look at it. And normally, the nuclear periphery acts as the graveyard fence. By keeping these transposable elements locked to the wall with H3K9E3, the cell keeps the ancient viruses permanently asleep. They can't copy themselves. They can't jump.
But when LBR and LAP2 fail...
Right. When they fail, and these regions float into the open active center of the nucleus, they lose those chemical locks. The ancient viruses wake up, and the very first thing they do is start transcribing their own RNA.
So they start printing their own photocopies. But how does the cell's internal security system know the difference between the RNA of a waking jumping gene and the normal RNA the cell uses to build its everyday proteins?
That is a great question. Comes down to the shape of the molecule.
The shape?
Yeah. Human cells typically produce single-stranded RNA. It's just a single ribbon of instructions. But viruses and these ancient jumping genes often produce double-stranded RNA, or these abnormal tangled RNA-DNA hybrids, as part of their copy and paste life cycle.
Oh, I see.
And the human cell has highly sensitive immune sensors floating around in the cytoplasm, specifically designed to hunt for double-stranded RNA.
Because if there is double-stranded RNA floating around, it almost always means a virus is actively replicating inside you.
Precisely. So the structural tethers fail. The jumping genes wake up and produce double-stranded RNA. The internal sensors detect this bizarrely shaped RNA, mistake it for a modern, invading virus, and pull the fire alarm.
Triggering that massive inflammatory interferon response.
Exactly.
That is absolutely mind-blowing. The call is coming from inside the house.
It really is.
The threat isn't external at all. It's the cell's own architectural failure, allowing its dormant viral ghosts to wake up, which then tricks its own immune system into attacking itself.
It's a profound demonstration of why genome architecture matters. I mean, we tend to think of the immune system as white blood cells patrolling the bloodstream.
Right, like an army.
But this shows that the physical spatial tethers inside the nucleus are an active structural immune defense system.
Okay, let's do a quick recap of that domino effect for the listener.
Good go.
Detachment of the DNA from the wall leads to the loss of chemical silencers. That wakes up ancient viruses, which produce double-stranded RNA, which then triggers a massive immune alarm.
That is the exact pathway.
This naturally makes me wonder about the broader implications for human health.
Oh, definitely.
If your cell loses this grip, your own DNA suddenly thinks it's under attack. Could some of the mysterious autoimmune conditions we see in medicine conditions, where the body chronically attacks its own healthy tissue for seemingly no reason, could they actually just be a structural failure inside the nucleus?
It is a highly compelling hypothesis that researchers are actively exploring right now.
What if it's just the LAP2 and LBR screws getting a little loose, causing a constant low-level viral alarm.
Yeah, the links between nuclear envelope defects, transposable element activation, and autoinflammatory diseases are becoming clearer every year.
That's incredible.
But the fallout of losing these tethers doesn't actually stop at immune activation.
There's more.
Oh, yeah. The ETH Zurich team found that it also completely destroys a cell's ability to know what it's supposed to be.
Yeah.
It causes a profound identity crisis.
Right, let's explore that final piece of the puzzle, because the researchers didn't just look at human cancer cells, they also looked at stem cells, specifically mouse embryonic stem cells.
Stem cells are the perfect model to study cellular identity because they're essentially blank slates.
Right.
They have this pluripotency, this potential to differentiate into any cell type in the entire body. During normal embryonic development, they make crucial decisions. They differentiate into 3 primary germ layers.
What are those layers?
So you have the ectoderm, which eventually becomes skin and nerves, the mesoderm, which becomes muscle, bone, and blood, and the endoderm, which becomes internal organs like the gut and lungs.
Okay, so to test the load-bearing walls here, the researchers engineered these mouse embryonic stem cells to completely lack those master screws from the very beginning.
Yes.
They knocked out the genes for LBR tempo, which again, for the listener, makes the LAP2 protein, and LNA.
Exactly.
What actually happens when you command these untethered, structurally compromised stem cells to grow up and differentiate?
Absolute biological chaos.
Really?
Yeah, the cells experience severe cellular amnesia. The researchers observed a massive deregulation of gene networks across the board.
Oh, wow.
Genes that were supposed to be turned on to build a specific tissue were silent,
Right.
and genes that were supposed to be permanently repressed were just blaring.
So they just failed.
Completely. The cells failed to form proper functioning cell types for the ectoderm, mesoderm, or endoderm. They were just stuck in this confused intermediate state.
Let me try out an analogy to see if I have the mechanics of this identity crisis right.
Let's hear it.
Think of the nuclear periphery, those walls of the library we talked about, as a memory vault, or maybe even better, like training wheels for cell identity.
Okay, now follow on.
For a blank slate stem cell to successfully become, say, a heart cell, it doesn't just need to read the instructions for making a heart. It has to permanently, irreversibly lock away the instructions for making a brain cell or a liver cell.
Yes.
It has to take those conflicting blueprints, shove them in the LAD bookshelves, and bolt them to the wall so they are never, ever read again.
You are absolutely on the right track. And it highlights a fundamental rule of biology, really. Differentiation is as much about turning things off as it is about turning things on.
Right.
A heart cell is a heart cell, precisely because it has silenced the rest of the genome.
But without LBR and LAP2, it can't lock those alternative instructions away. The shelves are broken.
Exactly.
So the brain cell blueprints and the liver cell blueprints keep floating back into the middle of the room. The cellular machinery is sitting there trying to read the heart blueprint to build muscle, but the brain blueprint keeps blowing across the table.
Yeah, it gets totally confused.
Tries to build a neuron and a muscle fiber at the same time and fails at both.
That is exactly the mechanism of the failure. This proves that the physical 3D organization of the genome, this specific process of tethering heterochromatin to the nuclear envelope, isn't just a convenient packaging solution.
It's vital.
It is an absolute prerequisite for cell fate determination. The researchers refer to this overall vital concept as preserving genome homeostasis.
Genome homeostasis, the active physical maintenance of a stable, functional genome.
Yes. And the key word there is active.
Right.
It is not a passive state where the DNA just sits there. It requires continuous, energy-intensive structural effort by membrane proteins like LBR and LAP2 to keep the chaos at bay.
To keep the jumping genes silenced.
Exactly. And to allow the cell to maintain a stable, singular identity over its entire lifespan.
So bringing this all together, we've gone from a simple structural question like how does DNA fit in the nucleus to uncovering a microscopic game of life and death.
We really have.
The researchers used intercepting RNA to shred the blueprints for the nuclear walls. They watched the restricted DNA detach and lose its chemical locks. They witnessed ancient viral ghosts wake up, tricking the cell's double-stranded RNA sensors into sounding a massive immune alarm.
Yeah.
And they saw how, without those structural tethers, a stem cell completely loses its mind, unable to remember if it's supposed to be skin, muscle, or bone.
It really highlights how interconnected the architecture of the cell is with its fundamental biology.
It's amazing.
Genome homeostasis isn't just an abstract concept buried in a molecular biology textbook. It is the active, physical process keeping every single cell in your body functioning correctly right this very second.
Right now.
Yeah, as you sit here listening, the microscopic walls inside your cells are actively bolting dangerous restricted DNA away from the machinery to keep you healthy.
Which leaves me with one final lingering thought for you to mull over.
Ooh.
If our genome is packed with these ancient, jumping, transposable elements just waiting for the structural hooks to fail so they can wake up, replicate, and trigger massive inflammation, think about what happens to our bodies as we age.
That's a fascinating angle.
Right. As we get older, our cellular machinery becomes less efficient, proteins degrade, structures weaken.
Right.
Could the general everyday wear and tear of our nuclear architecture, the slow, decades-long loosening of those LBR and LAP2 screws, be a hidden root cause of the aging process itself?
It's a very real possibility.
Are we essentially just slowly losing our grip on our own load-bearing walls, allowing chronic inflammation to take over?
It's a profound thought.
It completely changes how you view the architecture of your own DNA.